Now, if I took this structure and turned it so that you could look, let's say, in that direction, what you would see, this would be your gill bar, and then coming off in various places would be a gill ray. It's these gill rays, very thin toothpick-like structures, that are going to be supporting the soft tissue in between. So I just happen to make a cross-section, let's say right there, and that's what I'm drawing on the right. So it's a section that has a single gill ray coming off of the gill bar. We have soft tissue, and that's going to form what's called the interbranchial septum. Septum just means wall in Latin. It's always going to be an anatomical structure that separates something from something else. Inter means between, branch means gill. It's just the wall of tissue that's in between one gill pouch and the next gill pouch. So that would be this soft tissue. Sitting on top of that interbranchial septum are multiple gill lamellae. I didn't draw each one individually. There would be hundreds of these things all across the gill and on both sides usually. The gill lamellae are the site of gas exchange. So these are very thin-walled structures, often only one cell thick, so that we have a very short diffusion distance from the water to the bloodstream inside those lamellae. The combination of hundreds of these things, and often a shape like this, something like that. They have a complex shape. They're not just a cylinder sticking up. That complex shape, of course, gives it more surface area. Surface area is important because that enhances your ability to have diffusion occur to obtain the oxygen from the water. Ends up giving a very, very high surface area. Just some numbers I found somewhere. 50-pound sea bass has 60 square feet of respiratory surface. So yeah, that's a fairly good-sized fish, but still, that's a lot of surface area, 60 square feet. Okay, it doesn't matter what kind of fish you're looking at, you're generally going to have these structures, gill bars. Many times gill rakers are present. You're going to have gill lamellae because oxygen is going, and there's going to be interbranchial septum of some kind. We have different gill setups in the different kinds of fishes. So we have three major kinds of fishes. We have our agnathans, we have our elasmobranchs, our cartilaginous fishes, and then we have our bony fishes. So we're going to have three kinds of gill setups: pouched, septal, and opercular. Pouched are going to be in the agnathans. So this is inside the animal. That would be the pharynx. And then that would be on the outside of the animal. What's characteristic of pouched gills is we have a very thick interbranchial septum. This, where my hand is the interbranchial septum. it separates this gill pouch from this gill pouch. It's very thick in the agnathans. It would be supported by pieces of cartilage in there. It has a distinct pouch appearance, and you'll see in the other ones that it does not necessarily. Lining those pouches would be my gill lamellae, where the gas exchange is taking place. Note that I have a narrow opening leading into the pouch from inside the pharynx and a narrow opening leading to the outside. Just think of the gill slits on the outside of the lamprey when you looked at it. They weren't really slits, they're more like little circles. Right? But that's typical of pouched gills. Restricted or very small openings into and out of the pouched gills. The number of these, so pouched, the number of gill pouches in agnathans is highly variable, but it's generally larger than the other groups. So lamprey, for example, have 7 typically. Hagfish can have 6 to 15. Some of the ancient Ostrocoderms had 5 to 9. The point is there's a large number. That's typical of pouched gills. Septal is going to be characteristic of our elasmobranchs. So that when you draw this animal, this would be the inside of the animal. This is the outside. So the mouth is up here. This is the anterior. What we see in the elasmobranchs is that the interbranchial septum has become very flat and plate-like, right? Not thick like it is in the pouched gills found in agnathans. And in fact, the word elasmobranch means plate gill. Elasmo means plate, branch means gill. So we've taken that interbranchial septum and made it very, very thin. In addition, septal gills, fish with septal gills are characterized by having a very wide opening on the inside and a wide opening on the outside of the animal. Think of the sharks that you looked at in lab, okay? Very long slits on the side of those animals, so much larger opening than occurred in pouched gills. The number is somewhat variable, usually 5 to 7, right? So generally less than what we see in the pouched. Opercular is going to be what you find in the Osteichthyes. In these guys, the interbranchial septum is greatly reduced, and we get a V-shaped appearance to the gill. We get a V-shaped appearance to the gill, so our gill rays are branched. And the number of gills is reduced, so that most Osteichthyes, most fish that have opercular gills, have 4 pairs of gills, as opposed to the 5 to 7 in the elasmobranchs, and often greater than that in the agnathans that have the pouched gills. In addition, the structure is different in the sense that we don't have separate gill openings for each gill. We've reduced the number of gills, and we've reduced the opening to only 1. And that single opening is then covered by a bony flap called the operculum. So that's what this thing is right here. So these are some of the anatomical differences that we have among our fish. But they're all operating under similar principles. They're all using gill lamellae for diffusion of oxygen from the water into the bloodstream. Let's look a little bit in more detail how they work. This is true of the opercular. It's probably true of the others as well. One way in which fish can extract more oxygen from the water is by having a counter current system. So whenever you have a counter current system in biology, you're talking about two fluids that are flowing past each other, but in opposite directions. So a counter current. The two fluids, what are the two fluids going to be?
Blood and water.
Blood and water, sure. We're talking about getting oxygen from the water and into the blood. So we're going to have the blood moving in a direction opposite that of the blood. If that's my gill bar, and here's my gill ray branched coming out, and this is the hard part for me to draw. This is representing the gill filament on top of that supporting gill ray. So I'm drawing it in sort of like a half circle there. The setup in the fish is such that the water, let's say, would be coming from that direction, but the blood inside is flowing in the opposite direction. So when these fish breathe, and we're going to go over it in a little bit of detail in a minute, the water's coming in through the mouth. So you have the water coming in through the mouth and then the blood flowing forward. So the two things are going in opposite directions inside the gill lamellae, where gas exchange is going to take place. Well, why is that important? Let's illustrate without a countercurrent system. In other words, let's assume the two fluids are flowing in the same direction. All right, so my water is flowing in that direction, my blood is flowing in that direction. And let's just put down some arbitrary numbers. These are arbitrary units of oxygen that don't necessarily have to be milliliters or percents or anything, just arbitrary units. As that blood flows into the gill, it's going to start to pick up oxygen by diffusion. But this will occur. The water then is going to be losing oxygen to the blood as it flows past. If you have the two fluids flowing in the same direction, what's going to happen is that at some point along the length of that gill, we will stop having diffusion because the two fluids have equilibrated with each other. If we have a countercurrent system, so